Hello, Professor. In this opportunity, I want to give short presentation about cell disruption method, especially French press and homogenizer. Cell disruption is an essential part of biotechnology in the downstream processes related to the manufacturing of biological products. The disruption of cell is necessary for the extraction and retrieval of desired products. So many biotechnology products are from intracellular cell, must be released from cell before recovery. The chosen technology depends on the product, cell type, and scale. There is general method for cell disruption, such as mechanical and non-mechanical. Mechanical methods are those methods that require some sort of force to separate out intracellular protein without adding chemical or enzyme. In contrast to mechanical, uses methods other than force to rupture a cell's wall and membrane. For example, enzymatic lysis uses enzyme to disrupt the hydrophilic and hydrophobic bonds in the cell membrane. Detergent and chemicals work in a similar fashion. Different cells have different structures, hence they require different methods for disruption. Cell walls act as additional disruption deterrents, with this cell being particularly difficult to disrupt, as the cell wall limits the solvent's access to the desired product. In a French press or high pressure homogenization, the cell suspension is drawn through a valve into a pump cylinder, then it forced under pressure of up to 1500 bar through a narrow annular gap and discharge valve, where the pressure drops to atmospheric. By operating the press at higher pressures, the number of passes of the slurry through it can be decreased in order to obtain the degree of the disruption. However, the operating pressure may be limited due to the activation of certain heat-sensitive proteins, which may increase the number of the passages required. The French press is a small-scale method, whereas the homogenizer can be applied to a large-scale production. Homogenizers can vary in design and has a high amount of solids, up to 50% of the feed. Heat generation is also high, around minus 1.5 degrees Celsius per 1000 PCI. So as we can see, there are two equations between correlation about the correlation between protein release and number of passes, which is R is protein release first order with respect to the number of passes, which is N. The dependence of protein release on operating pressure that can range 400 to 600 bar can be expressed as a function of the pressure rise to an exponent. And the second equation is about correlation between protein available and pressure. The results can be combined in a correlation where R is protein release in kg protein per kg biomass, Rm is max protein available, K is rate constant and function of temperature, and A is the pressure of exponent. Okay, that's all. Thank you.